Right. What I was discussing is light dependent reaction and light independent reaction of photosynthesis. I'll give one little question which came from this section also. Light dependent reaction. What about the others? Shavindi, Gayani, are you all there? All right. Write the answers for these questions. Uh, here, here, here. These three questions. Quickly write the answers and tell me once you're done. Are you all done, children? Are you all finished? Are you finished with it? Okay, finish it, Sona. Hari, can we quickly discuss, Putte? Are you all done? Did you finish? Right. So, but then okay, this one, first one is asking, 1 second. First one is asking, what are the photosystems located? Sorry, where are the photosystems located in the chloroplast? What is the answer here? On the thylakoid membrane. In the exam, they took on grana also, but better term is on the thylakoid membrane of the chloroplast. What state the wavelengths of light absorbed by wait. State the wavelengths of light absorbed by chlorophyll A molecules in photosystem 1 and photosystem 2. Photosystem 1, it is P, sorry, it's 700 nanometers. Nanometer term should be there. PS2, 680 nanometers. Nanometer term should be there. State the three ways by the cyclic electron flow differs from linear electron flow. The question is not asking a comparison. You have to write about which electron flow here. Cyclic electron flow, not about the linear electron flow. Even if you have written like a comparison, you got the marks this time, but we don't know whether in future they will consider only one side. So cyclic electron flow, what we can say? Cyclic electron flow, only ATP, only word should be there, only ATP is produced. Water is not hydrolyzed. It or else you can say no splitting of water. Hydrolysis does not occur. And then oxygen is not released. You have to write not released part. Then instead of ATP is produce, only ATP is produced, you can say that NAD, sorry, NADPH is not produced. One of these, right? Occurs in PS1 only. Correct? Yes, that is also can be taken. Right, are you all done? Shall we move to the second part of this lesson? So here we'll be going to the second stage of photosynthesis, which is Calvin cycle, right? So We are going to the summary back. So we are going to Calvin cycle here. Calvin cycle. There are three stages. What are three stages, children, of the Calvin cycle? What are three stages, Pute? Carboxylation, else we can take it as carbon fixation. Next, what is the second step? Reduction and regeneration is the third step. And in this case, there's a little error in the book also. I'll prove you today with the Campbell biology book also. I'm just going to that book. So here, this is picture. This picture, since it come across, I will tell you. This is the thylakoid membrane of chloroplast. This is how photosystems are located on the thylakoid membrane of chloroplast. Here it is photosystem 2. Here it is photosystem 1. What we discussed early, this is happening like this. Likewise, there are so many photosystems located on the thylakoid membrane of the chloroplast. Right, so here it is Calvin cycle, children, having having cyclic nature. Initially, carbon dioxide fixation, or as we call it as carbon fixation. So here, carbon dioxide molecule is taken from RUBP, which is a 5-carbon compound. RUBP is taking 1 carbon dioxide. So 5 plus 1 is how many, Pute? 6 carbon. So here it is making a 6-carbon unstable compound. Another 6-carbon unstable compound. This is immediately broken down into 2 molecules of 3-PGA. So here 3-phosphoglycerate is 3-PGA, what we discussed in the lessons, right? 3-PGA. Got it? So it's soon broken down into 3-carbon compound, which is 3-PGA. 3-PGA is having 3 carbon in each. Likewise, 2 molecules are produced. Up to that, okay? So this is the actual carbon dioxide fixation, children. So we will be going to read what they have discussed in this one before we going to our one, okay? The Calvin cycle incorporates, can you see properly? Wait 1 second, I will increase the size. Right, Calvin cycle incorporates each carbon dioxide molecule one at a time by attaching it to a 5-carbon sugar named ribulose bisphosphate. It is also called as RUBP. The enzyme catalyzes this first step by RUBP carboxylase. The enzyme is Rubisco. This is the most abundant protein in chloroplast and is also thought to be most abundant protein on Earth. Right? The product of this reaction is a 6-carbon intermediate, so unstable, and it immediately splits in halves, forming two molecules of 3PGA for each carbon dioxide fixed. Can you understand? That part okay? I'm going to the book also one more time. So what they have discussed there, we'll see that is kind of a mess here. So here I'm coming to carbon fixation, CO2 fixation. The carbon dioxide acceptor is 5-carbon sugar, ribulose bisphosphate RUBP. The addition of carbon dioxide to RUBP is called carboxylation. The enzyme involved in this reaction is RUBP carboxylase oxygenase or Rubisco. So, first product of RUBP carboxylation is 6-carbon molecule which is unstable and breaks down immediately into 2 molecules of pyruvate. This is the first stable product in the photosynthesis. Children, remember up to this only carboxylation. And you can say that the enzyme is abundant. That part also we can take. But the last sentence definitely goes to this second stage, okay? There is a big error in the book. So they are not addressing that also. But in the marking, we asked from the sir. Sir said that it should go down, okay? Hi, I'll show you with the Campbell also. Tell me whether you understood the first step, the carbon dioxide fixation. Okay. I'm going to the second stage here in the picture. Right. Second stage, you can see that 3PG here. 3PG is converted into 1,3-bisphosphoglycerate using the ATP, some amount of ATP produced in the light-dependent reaction. It's energy demanding part that ATP energy is needed to produce 1,3-bisphosphoglycerate, and then thereafter only 1,3-bisphosphoglycerate is converted into glyceraldehyde 3-phosphate, which is again a 3-carbon compound. This is also 3-carbon compound. This 3-carbon compound into another 3-carbon T3P using the NADPH produced in the light-dependent reaction. The products from the light-dependent reaction is used in Calvin cycle here. The name Calvin cycle has come because many of the reactions of the Calvin cycle is found by a scientist called Calvin. It was say NADPH is converted into NADP plus and giving H pluses and electrons. Using that H pluses and electrons, 1,3-bisphosphoglycerate is converted into glyceraldehyde 3-phosphate. As you see, there is a color difference here. From here to here, this is the reduction phase, phase number 2, right? So I'm going to there. Reduction phase. We'll read from this and go to our one. Each molecule of 3-phosphoglycerate receives additional phosphate group from ATP, becoming 1,3-bisphosphoglycerate. You don't have to describe that much in detail. You can say that G3P is converted into 1,3-bisphosphoglycerate using the ATP, some amount of ATP produced in the light-dependent reaction. Next, a pair of electrons donated from NADPH reduces 1,3-bisphosphoglycerate, which also loses phosphate group, becoming glyceraldehyde 3-phosphate. Specifically, the electrons from NADPH reduces a carboxyl group on 1,3-bisphosphoglycerate to the acetaldehyde group of G3P, which stores more potential energy. G3P is a sugar, the name of 3-carbon sugar formed by glycolysis by the splitting of glucose. G3P glycolysis වලදීත් හැදෙනවා කියලා අපි ඒක ගැන ඉගෙන ගන්න නැහැ. අපි just intermediate එකක් කියලා දන්නවා glycolysis glyceraldehyde 3 phosphate. හැබැයි අපි මේකෙදි carbon dioxide හදන්න ගන්නේ G3P මම කියන්නම් ඊළඟට. Notice in that in the figure at every 3 molecules of CO2 that enter the cycle there are 6 molecules of G3P are produced. ඒ කියන්නේ මෙන්න මේ කතන්දරේ මෙතන 3 carbon dioxide to 3 ribulose phosphate producing 3 molecules of G3Ps here, right? 3 molecules of, 6 molecules of G3P here. Out of 6, 1 is going to produce carbohydrates and the other 5 is going to regenerate Rubisco. Sorry, RUBP, not Rubisco, RUBP. I'll explain this in my chart. Hari? Just we'll read it first. But only one molecule of this 3-carbon sugar can be counted as a net gain of carbohydrate. The cycle begins with 15 carbons worth of carbohydrates in the form of 3 molecules of 5-carbon sugar of RUBP. I think this part is complicated for you. I'll explain with my diagram, okay? Wait. All right. Here, right. RUBP there. RUBP, how many carbon, children? RUBP, how many carbon? It's 5-carbon compound, no? Yes. So it's 5 carbon here. This RUBP reacting with carbon dioxide. Carbon dioxide has 1 carbon, right? Isn't it? Right. So 1 carbon plus 5 carbon, how many carbon, children? It's 6 carbon, no? What is produced here? 6 carbon unstable compound, isn't it? 6 carbon unstable compound. So this reaction is catalyzed by which enzyme, children? Which enzyme? This reaction is catalyzed by which enzyme? Rubisco. Alright, enzyme Rubisco is catalyzing this reaction. Yes. This 6-carbon compound will be soon broken down into 2 molecules of 3PGA. හරිද? 3PGA having how many carbon, children? How many carbon are there in 3PGA? It's a 3-carbon compound. Correct? This 3-carbon compound is then converted to 1,3-bisphosphoglycerate. That part okay? What is used here? Wait, it's difficult to write letters like this. I don't have a mouse also here. Right. What molecule is used to convert this one, children? You tell me. ATPs are used, no? So here, ATP converted into ADP. ATP is taken from which reaction, children? Light-dependent reaction. Some of the ATP is utilized here. Is that part okay? ATP is now converted into ADP plus phosphate. And now this is converted into 1,3-bisphosphoglycerate. Then 1,3-bisphosphoglycerate is converted into G3P. That part okay? This is the reduction phase. The numbers is the problem now we had. They say that cycle should occur 3 times. If the cycle should occur 3 times, you should capture 3 RUBP. 3 into 5, 15. Here, 3 carbon dioxide should be captured. So, if it is like that, 3 into 15, so 3 into 5, 15, 15 plus 3, 18 is the total. 18 divided by 6, you will get 3 molecules of 6 carbon unstable product, and also you will get 6 of 3 PGA. That part okay? Up there, tell me whether it's okay. Alright. So this produces in return 6 of glyceraldehyde 3-phosphate and 6 of G3P. But I would say 5 of G3P is going to regenerate RUBP. Meanwhile, one of these are going to synthesize carbohydrates. That means glyceraldehyde 3-phosphate is the one which is going to synthesize carbohydrates. Correct? So here, one of these going to produce carbohydrates. Now we have a 3-carbon compound to produce carbohydrates. Now you should understand, in order to produce 1 glucose, you should produce 2 net G3Ps for carbohydrates. That means cycle should happen how many times to produce 1 glucose molecule? Cycle should occur 6 times. Do you agree? Netukita තේරුණාද? Here they are using NADPH produced in the light dependent reaction. It's being converted into NADP plus. Don't know whether you can see what I'm writing. Can you all see? Plus H pluses and electrons, right? So here also some of the ATP produced in the light dependent reaction is necessary. Remember very well, each and every reaction of this pathway is catalyzed by specific enzyme. There are so many intermediates. Even though we are not learning, it doesn't mean there are no intermediates. There are so many other names in this cycle. So we are not learning that one. Okay, so that remember, in order to convert each intermediate into the next intermediate, there's a specific enzyme. There are names for the enzymes too. So you have to remember is these are catalyzed by specific enzymes. When you're writing also, you have to mention that thing. Understood? How do I put it? Right now, everyone go to the book and read completely. The Calvin cycle completely. Take that sentence down. You can understand no, now the correct thing is that Kiela. Alright? Are you all done ready, Buddha? Right. Okay, Buddha. Now we are writing in the summary for the net synthesis of 1J3P. How many times the cycle should occur? How many times? 3 times. ඊළඟට for the net synthesis of 1 glucose, cycle should occur how many times? 6 times. Hi. ඊළඟට what is the first stable product of this pathway? What is the first stable product? 3-PGA. හරි නේද? Then what is the precursor? For carbohydrate synthesis? G3T. හරිනේ? Right. What else we can talk about this? What are the steps that use ATPs? What are the steps? Hmm? Oh, what are steps? Is it only regeneration? Reduction and regeneration. හරිනේ? Reduction and regeneration. ඊළඟට steps that use NADPH. What are the steps? Go. What are the steps that use NADPH? Ramai? Reduction. Yes. Okay, now? Hi. So usually the questions come to describe the Calvin cycle of photosynthesis likewise. So you can manage now. Are you all okay? So Maya, what happened to Rashida? Rashida told that she also have some problems in this section. What happened to Rashida? The class students, many of you promised that you will be coming, but only you are here. That's how usually happens, no? All the others said that they all are free. The Imasa is usually online students, no? Imasa and Gayunia, they are going usually not coming to our class time now. Karen, did you receive the photosynthesis too? Alright, okay, well, we are going to the next part. Are you all okay with this one? I'm going to this section which is about photorespiration. All right. The thing is, I'm repeating the same lesson because of these guys. These guys are not coming. I don't know what to say thing, right? So I don't actually have to repeat this one because I have covered this well. So here about photorespiration. Uh, Now this is like this. Right? Photorespiration is something like this. This Rubisco having the capability of catalyzing two reactions. One is it can catalyze carboxylation reaction as we know so far. Number one, RUBP mm, plus carbon dioxide will produce 6 carbon unstable and then soon produce 2 molecules of 3-PGA that is catalyzed by the Rubisco enzyme. So this is the carboxylation reaction of Rubisco. That part okay? This is called carboxylation reaction. Also, Rubisco can catalyze oxygenase reaction. Rubisco name itself, it is telling RUBP carboxylase oxygenase. RUBP carboxylase and oxygenase. Alright. So in the carboxylation, this reaction is happening. In the oxygenase, RUBP is taking oxygen and it will produce one molecule of 3-PGA and it's producing a 2-carbon compound. So here it is 2-3-PGA. Here it is 1-3-PGA. Usually what we need is to produce more and more glucose in this pathway now. So remember that in order to produce more glucose, more 3-PGA should be there. So the plants, the carboxylation reaction is preferred over oxygenase reaction. So the thing is, the say 3 plants having a disadvantage in the high temperature and high light intensity. They tend to close the stomata to prevent transpiration or reduce transpiration is the correct term. To reduce the transpiration, they close the stomata of the plant. Not every stomata, some of the stomata are closed. In that case, what happens is this stomata closed means they can't take more and more carbon dioxide, but in the light dependent reaction, more and more oxygen is produced, and in the substomatal cavity or in the leaf, oxygen is more now, carbon dioxide is reduced. Since carbon dioxide cannot be taken more and oxygen is continuously producing. So now this is the problem of having oxygenase reaction instead of carboxylase reaction, right? So they will prefer whatever the thing is in high concentration, right? So when the oxygen is higher, oxygenation reaction will happen. When the carbon dioxide is high, carboxylation reaction will be preferred. So at high light intensities and high temperature, oxygenation reaction will be preferred over carboxylation as oxygen and carbon dioxide are acting as competitive substrate. What are competitive substrate? Both of these can react with RUBP carboxylase enzyme, so that it's kind of a problem. Okay? Understood? And also remember, this will reduce the yield by 50% because 3%, so half percent of the PGR, 3 PGR produced here. So that is going to reduce the yield of the plant. That is one thing. And the other thing is this 2-carbon compound is highly toxic when it is accumulated in high quantity. So that you have to use an alternative pathway to convert this 2-carbon compound into carbon dioxide and water. Correct? So this is kind of problematic. Harder. So you need to use more and more enzymes to do this one. And also some of the ATP produced in the light dependent reactions because of this is because it is an energy demanding process. You need energy to convert this into non-toxic products. So that is kind of problematic. Respiration in the meaning says that this two carbon compound is converted into carbon dioxide and water. It's kind of similar to respiration, but it is energy demanding and it's kind of a wasteful process for the plant because enzymes are used in unnecessary pathway, meaning that those plants nitrogen use efficiency is low we compare to the C4 plants because enzymes are containing nitrogen, so that is used for unnecessary pathway. And also water use efficiency is also low in these plants because they can't manage the water amount and the carbohydrate synthesis at the same time. When they try to close the stomata and reduce the transpiration, they will be losing their productivity. Can you understand that part? Hi, now everyone go to the book and read about photorespiration completely and ask if you have anything you can't understand. You don't need to read from the computer. You can have it from your book and read it completely. Did you finish reading, Putte? ఎవరిది కేవలం? Right. Now, what's the condition, children? Is it understandable, or like you need more explanation regarding this cold respiration? This section is also usually comes in question papers, okay? So you have to remember this one. Right now, what's the condition? Now I did class from 8 o'clock. No, not 8 actually, 7 o'clock now. So if you're feeling really difficult, I can pause the class. Else I can continue and finish with the C4 as well. What is your request? Okay, Netu, Kim, I will explain the last paragraph. Mm, Others, meanwhile, tell your decision. Uh, Photo respiration is not only energy demanding, you know the reason why the ATP is utilized in the photo respiration pathway. No, that's why it is energy demanding. It's telling not only energy demanding, but furthermore leads to net loss of carbon dioxide, meaning that if you usually capture 3 carbon dioxide per cycle, you know that how many 80, how many 3 PG are produced now. So let's say you will capture 3 carbon dioxide. You know, in for that you will get 6 of 3 PG are produced usually in carboxylase reaction, right? But in this case, you know what? It's reducing the yield into 0.5%. You will get only 3 3-PGA molecules because of the photorespiration. So that you are capturing 3 out of that. Some of the carbon dioxide that you captured are totally wasted for a unneeded pathway, unnecessary pathway. Can you understand? Next one, it says, In each time Rubisco reacts with oxygen instead of carbon dioxide, plants make 50% less 3-PGA than it would have done if CO2 had been used. This potentially eliminates the net gain in photosynthetic carbon and lose productivity. All right, got it? That means if you are going to produce, let's say, 100 of something, now it is 50 of something. You are going to produce 50% less yield than requested. Okay, right. So what is now? What is your decision? Can I go to C4 or what? Hari will do like this. What about others? Hari will take like a 10 minutes break. I'll break the recording also here. So we'll discuss C4 in a little while. Take 10 minutes break and come back again. Okay.